Hi all. Today I'm excited to be introducing Dr. Yuju Chen. Dr. Chen is coming to us from Academia Sinica, and she is also an adjunct professor at many universities in Taiwan. Today she will be presenting to us on nanoscale to single cell phosphoproteomics. Welcome, Dr. Chen.
Can you hear me now? Hey, can you hear me in the back? Okay. Thank you very much for the invitation. We are kind of newcomer in the field of single-cell proteomics, and my lab is interested in clinical proteomics for long term, and accidentally entered the single-cell proteomics field. So today I'm going to share with you our very recent study on the single-cell phosphoproteomics, and the story is it? Okay, so the story starts with our very activities on the protein modification, especially on the cell membrane. And I started to go into the proteomics when I know nothing about biochemistry. So at that time, I thought, oh, cell membrane must be very important. But I did not realize it's so challenging. So if you look at the cell membrane, on the outside of the membrane, we have a lot of extensive modification on the glycan structure. And because of unique structures, so that is important for a lot of affinity between cell-cell communication or the protein-protein interaction. But after the cell receives the signal, the next step is signal transduction. And it's interesting to see that they are coming from the same protein. So there's a lot of interesting questions remain. For example, on the same protein, whether the glycosylation may also change the protein phosphorylation. But unfortunately, due to the technical challenge, most people still work on a single modification, either glycosylation or phosphorylation. So a lot of questions actually still remain. For example, I'm so interested to see what is the site-specific regulation. This may be extremely challenging for even glycoproteomics. And we talk about signal transduction. What is the absolute amount of the quantitation that is achieved or essential to activate the cellular function? This is still not known. And finally, I'm also interested in the synergy and dynamics of this kind of modification. So far, we have a lot of biomarker, we have a lot of target therapy using receptors. But even for the case of EGFR, for example, one of the most popular target therapy, there's a lot of unknown questions. For example, what are the signal transduction of EGFR for the patient level? And not to mention a single cell level. So I think this is a great space that we still need a lot of effort to understand what is happening on the cell membrane. And we actually spent some years to learn the technology, to develop technology required to answer these questions. And this is a kind of an old story we learned from nature. And my best friend, Guo Yiling, is a very excellent B-cell biologist. And one day she told me that she found something interesting. When the B cells start to have accumulation of ocucination, she discovered that the B cell will go from activation to apoptosis just by accumulated ocucination. So we perform very extensive dynamic phosphoproteomics and also the ocucination to analyze what is happening at the cellular level. To make the story short, and finally, we discovered that the single modification on LSP1 will change the B cell from activation to apoptosis. So once the B cell receives ocucination, and that will further recruit other kinases like PKC on the LSP1. And the PKC is a kinase to further do phosphorylation on LSP1. So it's kind of amazing that by two modifications on the same protein, the cell fate is totally changed for the B cell. So, the nature is so amazing. And encouraged by this story, we further pursue whether this is also helpful at the patient level. And this is an example on the CLL patient analysis. We try to analyze the phosphoprotein profile of the B cell purified from the human, from the CLL patient, and compare to the healthy control. And one of the protein we discovered is CAP1. Is block of layer, serine phosphorylation on the 473. And we spent a few years, 2 to 3 years, from cell line all the way to the animal, and discovered that this is an important site to regulate cell cycle progression for the CLL. And then I'm much more interested to see whether this is useful. And by patient analysis, we discovered that this specific phosphorylation site is very, very dependent on the inhibition of the drug and also recurrence of the drug. But during the process, you can see that we just recently published, but this project started, I think, more than 5 years ago. The challenge is it's so difficult to analyze the phosphoproteomic profile from the limited cell. And even we collect the sample from the patients, there are only a few micrograms of the sample. That is so challenging to see sufficient depth of the phosphoproteomics. So even this is useful, we realize the sensitivity may not be good enough for the clinical proteomics. So we start to see whether we can try other approach. And then we start to be very interested on the data independent DIM mass spectrometry. So we come up with the idea, if we are able to collect a big library using different kind of cell line, different kind of tissue from the same disease, maybe we can use this kind of global spectral library to enhance the sensitivity with a single shot manner. And this was done many years ago. So at that time, we are so excited that we are able to get many thousands of the protein and many thousands of phosphopeptide in a single shot DI approach. So with this success, you can also see that it works so well, even for the phosphoproteomics. So we use 165 synthetic phosphopeptide from the non-small cell lung cancer pathway. And you could see that the quantitation performance is excellent. So encouraged by this approach, we start to see, oh, DIA is a wonderful tool. But then we encounter another challenge again. From the cell to the animal to the human tissue, even for FFP tissue or biopsy tissue, everything is perfect. But when you come to the even more challenging sample, like for example, the tumor microenvironment, the cell-cell, immune cell, cancer cell interaction, you need much, much better sensitivity. But I also want to comment that when we count the protein identification number, it really depends on the depth of the disease or the biology. You cannot tell, oh, we must need 1,000 protein, 2,000 protein. The truth is we have to see the core of the network of the specific biology or disease. So this further inspires to explore the possibility to see whether we can have more sensitive approach for the clinical proteomics. And again, so that comes to the discussion about the current state sensitivity. And recently, we just published a review article. We try to summarize all the sensitive approach from the community. So the blue line, the blue circles means the single cell proteomics. And you can see that there's a lot of advancement for the single microscale all the way to the single cell proteomics. And I think a lot of inventions are from the experts sitting here. But if you look at the pink circles, which are the phosphoproteomics, the base approach is only 100 cell level. And this was published by Dr. Jiafeng Cai, who is also my former student. But this is the beautiful and nice work he published in the PNNL. So you could see that compared to the rapid advancement of the single-cell proteomics, the phosphoproteomics still lag behind because the total abundance of the phosphorylation is so low abundant compared to the peptide, the free peptide. And there's a lot of challenge, not only the low abundance, and it's also very dynamic in the case of the protein phosphorylation. And the phosphopeptide is more negative charge compared to the free peptide. So overall, there's a lot of challenge associated with a single cell level. And so we start to think whether we can have a better approach to take care of this. And then I met my colleague, Dr. Xiaolin Tu, who is the expert for microfluidic device. And so we thought that maybe this is an approach that can be fully automated to minimize the sample loss with a lot of flexibility. So for example, this microfluidic device can be, the battery is low. Is that okay? The battery is low. Okay, and so with this micro device, you can have a very simple, flexible design for different kind of configuration. And in the beginning, we can even do cell culture. We can do drug screening method. You can use a microscope for everything. And following the proteomics approach, we can also design different kind of material, for example, functionalized nanomaterial, titanium oxide bead, to do the enrichment for different kind of protein modification. So we thought that maybe this is the way to go for the modified protein analysis. And then we started to test the microfluidic device for the single cell proteomics. But initially, we actually designed this for patient analysis. I thought maybe this is nice for fully automatic analysis. So we started from 5,000 cell, 1,000, and finally we realized, oh, that's great for single cell. That's how it started. And you could see that even at a single cell resolution, it's very, very nice to see that we are able to do observe the EGFR signaling pathway in a very comprehensive manner. This is really, really amazing. But then when we want to test this on the single cell phosphoproteomics, and not to mention single cell glycoproteomics, we encounter a lot of challenge. Compared to the single cell proteomics, the phosphoproteomics or other PTM usually require enrichment of the modified peptide for better coverage. So if you look at the conventional workflow, there are several steps that we are not able to use the microfluidic device. For example, to do the cell lysis for trypsin digestion, sometimes we need to do the desalting, we need to change the buffer, and we are also not able to do the centrifugation by microfluidic device. So that means we have to modify the process. So first of all, we test any reagent that can be used for lysis digestion in the same tube without any desalting. So we compare the 4 types of the reagent, and we find out that SDC is actually working perfectly with the microfluidic device. And we further find out that the digestion time can be further reduced to only 1 to 2 hours. This is very complete without any missing value. And finally, we also add the DDM. This is suggested by Dr. Jiafeng Cai in his paper to minimize the non-specific absorption on the tube. So after the protocol is optimized from the micro scale, you could see that the coverage actually improved a lot. And following this process, the whole process can be completed in 2 hours, very, very fast. And we also use the library, DRA library, to improve the sensitivity of the coverage. And you could see that we actually generate different kind of library. The small library means that the size to construct the library is comparable to your sample. Because when you do the DIA library search, the peptide distribution in the retention time domain should be very similar. That can give you better identification coverage. And in the large library, even you have much higher number of the peptide precursor. But remember that they are in the different complexity in terms of the peptide composition and also the interference from other peptide. So we call spectral match, sample size comparable library. So from this, you could see that at the microgram level, you are able to actually see beautiful biology. You are able to see the depths of the biology very easily. And we just published these papers, and this is a very simple method using single tube. And the protocol is extremely easy, and you are welcome to check out the protocol for up to some microgram of the sample. So we adapt the whole protocol to the microfluid device.
Hey, Yuju, you asked if there were any questions. So,
Okay.
I may have missed, I think I did miss it, but so you are doing enrichment or you are not doing enrichment?
We are doing enrichment.
Okay.
Yeah.
Do you think there is a point at which you lose more from enrichment than you gain, like at the single cell level? Because you don't get perfect recovery when you enrich, right? So do you think there's kind of a sample size where enrichment no longer makes sense?
Yes, this is a great question. We actually compare the enriched sample and without enrichment. And actually, a few years ago, there's a group published a paper. They used a few hundred samples to search the single-cell phosphopeptide. And they are able to get like hundreds of the phosphopeptide from hundreds of the single-cell data. But based on our enriched sample, with the Eclipse, we don't have the extra yet or the timsTOF yet. So based on Eclipse data, we are able to get few hundreds of phosphopeptide at a single cell level. So I do believe that enrichment is much, much better. But if you talk about the nanoscale or the microscale, there's a huge difference. So enrichment is still very important. And in the case of the glycopeptide, it's the same. Say if we have 1 microgram of the sample, the serum sample, we are able to get like thousands of 1,000 of the glycopeptide. But if you do not do any enrichment, then you probably get much, much less. But this is based on the Eclipse data, Lumos data. And I think now we have much faster speed of the mass spectrometer like Ultra or the timsTOF. So that might be another level. Yeah.
No, thank you very much. And is there, since most of us don't have access to the microfluidic devices, right? Is there an alternative workflow like phage tip or something like that that you could do instead for enrichment?
Okay, for the one-part protocol I just mentioned, for the phosphopeptide level, we are able to get like 0.2 microgram. So that's few thousands of the cell. But for the protein level, it can be go down to hundreds. But we are not able to get even more. So I think the microfluid device has the advantage of the sample loss, minimal sample loss. Yeah.
Thank you.
Thank you. To follow up on Ryan's question, you started talking about the interplay of phosphopeptides and the signaling through glycopeptides to phosphopeptides and so on. Did you also use any enrichment tools for glycopeptides or glycans? Maybe you didn't get to that yet.
Great question. Great question. If you asked me the same question 2 years ago, I will tell you no, different material. Glycol and phosphopeptide are both negative, but they have the different hydrophobicity and also different degree of the negative charge. So it's kind of difficult to use the same material to do the enrichment. But recently we have work using something called Trilic, which is a hydrophilic material for conventionally used for the glycopeptide. And we use that for glycopeptide for some years, but we have difficulty to get highly purified phosphopeptide because of the low hydrophobicity of the phosphopeptide. But recently we discovered that if we add the metal ions into the Trilic material, that material alone can be used to enrich both glycol and the phosphopeptide in a sequential manner from the same sample. So this is one of the methods that can enrich both. Yeah.
Did I understand correctly? You used helic?
Trilic. It's not helic.
Not helic?
Are you a chemist?
Yeah.
Okay, great. For helic material, there's amine and also phosphate. Okay, and in helic material, the positive charge is inside, and negative charge is outside. So if you have a bit and positive charge inside, that means your negative peptide has to be going to inner side to do the interaction, to do the binding. So the efficiency is not good. But for trilling material, the polarity is switched. So you have the positive charge outside to extract the glycopeptide or the phosphopeptide. So that has much better efficiency compared to the helic material. It's called trillic, C helic.
Oh, okay.
Yeah, it's ionic reagent. Yeah, so we just spike iron into the trillic, then it can be used for enrichment of the both glyco and phosphopeptide.
Thanks.
Yeah. Yeah. Or I can use my computer.
I'm wondering if you have worked with, for example, high pH fractionated libraries.
Where's the microphone?
Here.
Oh, okay.
I'm,
Sorry.
I'm wondering if you have tried, for example, to work with high pH fractionated libraries of bulk samples that have been phosphopeptide enriched to help like a little bit the DIA software to find more IDs.
So can you rephrase your question?
So when you have like a library,
Yeah.
a phosphopeptide enriched sample, like let's say a bulk sample, yeah, use that as reference for the search with the software in single cell samples.
Yeah.
If we try that out to increase or improve the hits.
Yes, yes, that is actually in my talk. This is actually a very important question. When we work on the single cell proteomics, the library is not an issue. You just try to, like Alex mentioned, you need to have a high quality of the spectral library, and that can enhance the single cell proteomics. But when we work on the single cell phosphoproteomics, we encounter the challenge that I don't know if I can use the name. Okay, spectronaut. Even we pass all the FDR control of the spectronaut, and you find out that some of the peptides do not have the good quality on the mass spectra. You know, usually we will check the MS fragmentation, the number of the fragment ion, whether the phosphosite can be located for site determination. And we find out that some of the low abundant peptide, phosphopeptide has very low number of the fragment ions, but they were still identified by spectronaut. That's why we also consult Alexi to use the FreePy to confirm this part. So I think for this kind of modified peptide, the computational tool still need a lot of space to further improve the confidence of the identification. Yeah. Okay. Thank you. Okay, so we adapt the protocol into the microfluidic device, and this is a very simple concept. So you do the titanium oxide first, and the reason that we use the titanium oxide because it is a bit material. It can be easily packed into the column, and then we do the cell captures. It's all fully automatic, and we also have a microscope to look at the imaging of the size or in the morphology of the cell, and you can do the cell lysis in the same chamber together with the protein digestion. So after everything is done, is ready for mass spectrometry. You don't need any desorting at all. So that, because this is a simple step, so that will minimize the sample loss for single cell phosphoproteomic data. So as you can see from here, one of the advantage of the food device is you are able to adapt any kind of accessory to the chip. So we can use the microscope to look at the real image of the single cell. You can also count the cell. And one of the revision compared to our previous version is that the cell utility efficacy is very high. That means you don't need to have tens or hundreds of cells flowing into the chip. You just need to the very small very small number of the cells flowing to the chip. It's nearly 100% of the capturing efficiency. So that is very nice for the limited sample. And following that, we use the DIA library-based DIA to improve the coverage. And so this slide explains why I mean the sample size comparable library. So on the left-hand side, you can see that we actually generate different sizes of the library using different amount of the sample. And then when you use the data set to test the different library, you could see that for the smaller cell number, the small library actually improved the coverage because of the similar peptide space in the LC-MS/MS domain. But when you have the higher cell number, the larger library is important. And sometimes people ask me, how do you know which library works better? Actually, we find out that if you use the sample amount like 2 to 5-fold higher, that usually is the best. But it still depends on the sample type. So I think this is a very important point of building the spectral library. And so it's kind of amazing that from 10 cell to 1,000 cell, I forgot to mention. So even with the 1,000 cell, you could see that we are able to identify more than 10,000 phosphopeptides. That is already sufficient for a lot of biology of your interest. And even with the 1,000 cell, we are able to see many, many kinases. And also, you can see the very comprehensive coverage of the non-small cell lung cancer pathway. So for example, for EGFR, the tyrosine site, these two tyrosine sites are the so-called autophosphorylation site. That is the indication of the EGFR mutation, the driver mutation. So at the 10 cell level, it's amazing that we are able to see that. But after 10 cell, then we start to have the challenge for the single cell phosphoproteomics. So this is the one I just mentioned. We use the spectrum now to do the library search. But even everything past the confidence level and FDR control, you can still see that a smaller group of the peptide is in the low range of the normal distribution. So that suggests that this phosphopeptide may not have the good quality, or at least the confidence is not high enough. So we're struggling this for a lot of time. And then we find out that we probably need to do a more stringent control on the confidence of the phosphopeptide identification. So we follow the, first of all, we follow the monodistributed distribution of the precursor. We make sure the precursor is correct. And we also use the higher cell number to verify if the phosphopeptide pattern is correct. So by using this, then we reduce some number, but we want to make sure all the identified phosphopeptides are highly confident. So I think this is still issues that we really need help from the informatics expert. And thanks to Alexi and Fang Qiao, the great student and now postdoc there. And we also use the FLAPI to confirm the identification of the single cell level. So you are able to see that we have a few hundreds, not high yet, but few hundreds of the phosphopeptide at the single cell level. It's kind of amazing that even at such a smaller, very low coverage, we still see a lot of phosphopeptide that have been reported in the literature related to cancer biology or the drug resistance, etc. So I think a lot of people said that the important molecule must be low abundant. But in the case of the single phosphoproteomics, we may not necessarily agree with this kind of argument. We see a lot of phosphopeptide, important phosphopeptide are still there. That means they are in the basal level. But with such a low abundance, we really want to see whether that is likely useful. So we tested on a very interesting and important question in Asia. EGFR is high percentage in Asia countries. So our government have a lot of money paid to the reimbursement of the target therapy, all free for our patient. But eventually, nearly every patient will develop resistance to the EGFR target therapy. And then they can only perform chemotherapy. So the question is, for this dying patient in the late stage, is it possible that we are able to suggest any potential drug as a next line treatment for this dying patient? So we thought that most of the target therapy are kinase. So phosphoproteomics should be a good approach. So we tested this analysis for 3 patients from the stage 4, and they already develop resistance to the third generation EGFR target therapy. So with 10 cells, we are able to see about 1,500 phosphopeptide. And it's very amazing that with such a low coverage, we actually observe the biomarkers for the two most abundant lung cancer cell type, adenocarcinoma and SCCC. And these three patients are actually from the same clinical staging and same EGFR mutation. No difference at all in the clinical diagnosis. But we observed that this specific patient, 157, has the biomarker for HCC, but clinically, this person was diagnosed as the ADC cell type. So we went back to the hospital and consulted my collaborators whether there's a wrong annotation or something is wrong there. And then they discovered that this patient actually has a very low amount of the SCCC composition. So I think this example demonstrates that this kind of cell heterogeneity was not able to be detected by the clinical tools. So this also highlights the importance of the small cell population analysis at the patient level. And for these three patients, we actually see a lot of difference between their phosphoproteomic profile. So for example, this female patient has the activation of the containing complex, especially the YAP1, which has been recognized as an important regulator for the drug resistance. So finally, we want to see whether this kind of analysis is useful. And this is not single cell level yet. This is the 10 cell level. We are able to see that two phosphorylation sites on the EGFR are activated. And we also see the phosphorylation site for the ERBB2, the HER2 therapy. So we talked to my colleague whether he is able to test the drug for the patient. So he used the primary cell from the same patients from the lung pleural infection, and he confirmed that the drug obtainable targeting both the EGFR and the ERBB2 is useful for this specific patient. So we are very happy to see that this kind of research assay is likely be useful for the patient to indicate the potential next-line drug treatment for the future. So I think this, again, highlight the potential utilities for this research. But unfortunately, we don't have the most high-end mass spectrometer, so the throughput is really an issue for our current research. And we hope that we have the new mass spectrometer coming later this year to really enhance the throughput. And finally, I think we are very happy to make our dream come true for the single-cell phosphoproteomics study. And even though we achieved the first goal, but I think there's still a lot of challenge associated with this kind of single-cell protein modification. And the data deconvolution from DIA, I think, is still an issue. And the site localization is still very, very challenging, given the very low number of the fragmentation pattern for a lot of phosphopeptide. But with this approach, I think we are able to ask more interesting questions. For example, I think we start to have the tool to ask how does the site-specific regulation at the individual cell level. And we may be also to start to ask the single-cell signaling issue, whether the site-specific phosphorylation occurs on the same cell or different cell. Because what we observe from the past is the accumulation of the overall result. So I think this likely open another field. And I also talked to my student. When I tell him to do the project, he was angry for 1 year, and he always argued with me because this is something impossible. But now I think maybe it's time that we can pursue like single-cell glycoproteomics, but we still need to demonstrate the utility of this kind of research. And finally, I want to thank my colleague. Dr. Tu is the expert to make the microfluid device. So if you are interested, you can talk to him. And Taiwan kind of empire of the semiconductor industry. So chip production is kind of easy and very, very cheap for our countries. And we are hoping to really push the tools for the practical utility in the future. And finally, I also want to thank the patient. And clinically, there's a lot of questions that might be useful for proteomics. So I'm hoping that we are able to really push the utility for the patient in the future. Thank you very much for your comment.
Thanks. Thank you, Dr. Chen. So I think that we have time to take one question. So I will here.
Yeah, so I'm a little bit interested in the microfluidic system for single cell sample preparation.
Mhm.
I mean, I'm an old microfluidics engineer, actually. I did that early in my PhD. So how much of an issue is like bubble formation? Because I guess that is usually a problem, that bubbles get in and they get stuck and they can.
How to start to get it started?
Yeah, how do you deal with bubbles? And
I don't have the slide to show. That is actually the most challenging question in the beginning. So my colleague, Dr. Tu, used a high pressure to push out the bubble, and also the surface, the design of the size of the channel is also highly important. You have to measure the fluid mechanics, is in the most optimal condition. So you can check out our design. And I think he has all the detail in the publication. Another thing is the surface chemistry. You have to make sure that after you do everything in the clean room, everything on the surface has to be very clean. Otherwise, you introduce a lot of detergent reagents into the mass spectrometry, so the data will not be good. Yeah, so I think these two are most critical.
can I ask just one small question? Or okay. I mean, of course, when you prepare on a tip, this is something you can dispose of afterwards. Are these microfluidic systems, are they reusable? Like, how do you clean them
yes.
efficiently so that you can load a new set of single cells and make sure that you're not contaminating?
Yes, we tested the chip for, I think at the current stage, the base utility is like 5 times. But because it's so cheap, so we don't usually reuse the chip.
Okay.
Yeah.
Oh, thank you.
Thank you.